Okay. So first off, thanks to Dave for the invitation. I'm delighted to be here to present some of our preclinical and basic research on traumatic brain injury outcomes and the chronic effects of brain injury, in particular focusing on brain neuroinflammation, but also effects outside of the brain in the periphery. So depending on the severity of injury, the physical impact to the brain can cause meningeal injury, axonal injury, or vascular damage that can initiate a series of injury events mediated by damage associated with molecular patterns. Sorry, I just want to add this, be a little bit easier. DAMPs that can initiate activation of glial cells, neurons, and neuronal damage. And this puts into motion secondary injury events initiated by that may be related to mitochondrial dysfunction, glutamatic cytotoxicity, and production of cytokines in reactive oxygen species. But then drives these additional injury events, which is a call to the periphery to recruit leukocytes. There may be blood-brain barrier disruption, and also edema formation that can lead to increase in intracranial pressure. Now, depending on the severity of injury and the type, these peripheral recruitment of these immune cells, as well as the activation and work of the resident microglial cells, can try to mediate the protective clearance response that may also lead to tissue repair and regeneration ultimately. But depending on the injury of the severity of the injury or the chronicity of the injury, these protective effects may become damaging, leading to these chronic inflammatory events that are driven by neuroinflammation through this feed-forward loop. And it's seen that when under these chronic injury events that the neuroinflammation that may lead to neuronal damage, neurodegeneration, actually participates in some of these long-term neurological dysfunction and neurobehavioral deficits seen in patients. So this is the experimental framework that we're thinking of, but the clinical data that support this is actually led by Dave's group at Imperial and other groups that are looking at patient cohorts that survive beyond 1 year. And in this nice study published in 2011, Dave's team identified that at the level of the thalamus, these are T1 images of the thalamus, and what's shown here is the PET imaging marker for neuroinflammation, PK1195, overlay onto these T1 images, and what in these severely injured population at 204 months or 17 years post-injury, compared to the age-matched control, you can see increased binding of this inflammatory PET ligand, which was most evident in these injured populations of the putamen and thalamus. This was associated with other atrophy of the brain in the hippocampus and other regions, as well as loss of executive functioning within this population. So severe injury can lead to long-term neuroinflammation. And when populations come to pass and come to autopsy, other work by Willie Stewart at Glasgow have demonstrated in these cohorts of subjects, approximately 30% of individuals who survive beyond 1 year can have chronic inflammation, particularly at the level of white matter. And this is an example of a 37-year-old male surviving 4 years post the single severe TBI. And what you'll notice here is a lot of these reactive microglia that are associated with a thinning of the white matter within this population. So we use this type of research to stimulate our basic principles, and that's essentially what I'd like to show today is the modeling data that we're involved in. So to model severe injury, we use this cortical contusion injury model, which is a focal contusion. It's an ipsilateral injury induced on the left parietal cortex, and it drives this primary injury, which then initiates secondary injury events. And looking at microglia, you can see, particularly in these two-photon imaging, there's a dramatic change in the function of the microglial cells in responding to injury, such that they change their morphology and their transcriptome and all these other things to respond to injury. They alter their phenotype and become more phagocytic, have a larger cell body and reduced processes. And when I set up my lab several years ago, we wanted to quantify this response over time, and particularly in the chronic phase. So we identified and quantified in the cortex, hippocampus, and thalamus these number of these hypertrophic or bushing microglia. And as expected, there was an acute injury event. The peak of the inflammatory response was at 1 week post-injury. But when we followed cohorts of animals up to 1 year post-injury, these reactive microglia were still seen in the brain. And this very much correlated with hippocampal and cortical neurodegeneration, and also behavioral deficits, including motor and cognitive deficits in these animals. When a talented postdoc joined our group, Rodney Ritzel, he applied his flow cytometry technologies to explore and to try and understand what was happening in these microglial cells at this chronic injury time point. This is a snapshot of what's happening at 90 days post-injury in microglia. And what his studies and additional studies have shown is that microglia are very reactive. They're producing a lot of cytokine, TNF, IL-1, reactive oxygen species. But they're also showing markers of immune senescence or senescence and dysfunction, such that at 90 days post-injury, they have higher levels of P2HX and BCL2, two principal senescent markers. This suggested that they have impairments in function, including phagocytic function, which is their critical function when they play a role in removal and garbage collection within the brain. And such in these ex vivo assays, when Rodney presented these E. coli particles, but also in additional studies, myelin protein that they would see in the injured brain, there was impairments, such they had functional impairments in functional capacity. So this is a very interesting chronic injury event that we thought that was worth investigating further. And we decided to use this drug, which is called, it's a CSF1 or colony stimulating factor 1 receptor inhibitor. These are very popular in neuroinflammation at the moment, particularly after the work of Kim Green at UC Irvine, which shown that these type of drugs are really useful tools for depleting microglia in the context of the brain. So CSF1 is required for microglial survival, such that if you use an inhibitor, and it's shown here, you can remove individual microglia when this inhibitor is given in the food, in the chow given to animals. Interestingly enough, in the absence of microglia, these animals have normal cognitive function. But also, what's really interesting is that there's a 5% population that are resistant to this inhibition by this CSF1 inhibitor. And when you remove the drug from the system, you actually repopulate or retile the brain from those resistant cells. So this is, again, a very interesting tool that we can study microglial function in the conditions of pathology, but also the repopulation, the rejuvenation of these microglia after an injury event. And this has been done in the acute phase very much. So what we thought to do was ask the question, during chronic phase, would it be possible to remove those chronically activated microglia and repopulate them with another population that may be less inflammatory? And what would be the impact on motor and cognitive function recovery? This was the project taken up by Rebecca Henry, a postdoc in the lab. And what she chose to do is injure cohorts of animals using the CCI model. And then at 4 weeks post-injury in the chronic phase, she administered cohorts of animals to this PLX treatment, which removed the microglia, which is shown here by these flow cytometry plots or in the histological approach. And then after a week, she returned the animals to the normal chow. And that resulted in a repopulation of the microglia, okay, within that group. Then at more later time points, she looked at behavior, motor and cognitive function, as well as molecular and cellular pathways. And what was most striking with these motor function tasks was there was improvement in motor function recovery in those animals that received the PLX treatment. So let me just take you through this data. So all animals after TBI have impairments in motor function, as demonstrated by this BMOR task. So such that at 4 weeks post-injury, when we administered this drug, they all have the same level of motor impairment. But after the removal of the reactive microglia and the repopulation with alternative forms of microglia, there seems to be this recovery period where there's improved motor performance through 12 weeks post-injury in comparison to the vehicle-treated cohort. When we assessed cognitive function using the Morris water maze, similarly, we saw there was a change or difference in their ability to learn this memory task, the traditional Morris water maze, such that they had reduced latencies in the acquisition phase compared to the injured with vehicle treatment. And in these probe trials or search strategy trials, what you'll notice is that the vehicle-treated animals spent less time in the target quadrants during this recall test, or they took a longer time to find the missing platform in this test. But those animals that received the PLX treatment, which had the depletion and repopulation, actually had improved performance, indicating improved cognitive function and retention memory. Furthermore, there was reduced lesion size, as shown in this neuropathological assessment. But also, when we looked at the molecular level, there was a change in the inflammatory phenotype. So what Rebecca did was perform some nanostring analysis to look at the transcriptome. And what she showed was that TBI induces this very reactive inflammatory profile, with a lot of the markers that we would see in chronic injury events and chronic neurodegeneration, including NLRP3 inflammasome, complement system, and NOX2. There are also some interesting and important changes in oxidative stress and antioxidant responses. And when Rebecca drilled down a little bit further on this, she wanted to investigate the NOX2 NLRP3 inflammatory axis, which we'd been studying before. So there is this relationship between reactive oxygen species and priming of the inflammasome. And what she had shown was that the PLX-treated animals had less of this reactive NOX2 protein within microglia at 3 months post-injury. In addition, and to support this concept that this pathway was targeted, when she evaluated NLRP3 inflammasome, there was also a reduced expression of this complex within the cortex. And when she looked at microglia specifically, there was a reduced cleave caspase 1 and IL-1 production, which is the product of the inflammasome in those microglial cells, suggesting that this is indeed an important target of this delayed treatment. So these studies indicate that, the severe level injury causes motor and cognitive dysfunction, which is driven in part by chronic reactive microgliosis through this NLRP3 NOX2 pathway. And this may lead to chronic neurodegeneration and cognitive decline. It also indicates that with these delayed treatments, if you remove this reactive microgliosis from the system at 1 month post-injury, you can actually improve the cognitive performance in these animals and also reduce the inflammatory burden within the system, such that the chronic phase neurodegeneration is arrested or halted to some extent. And we believe this has important indications for chronic neurodegeneration and also the therapeutic window, so that the therapeutic window may be much longer than previously considered. But TBI also causes effects outside of the brain, peripheral effects, which cause immunosuppression and also immune dysregulation. And these are driven in part through the autonomic nervous system, by mediating dysautonomia, through the HPA axis by glucocorticoids and catecholamine release, but also through cellular defense responses. And particularly in the setting of acute injury, immunosuppression increases the susceptibility to ventilator-acquired pneumonia, which is a major killer within the acute ICU setting. So what we have considered over this last while is what's the effect of chronic injury and these secondary challenges, secondary inflammatory or respiratory challenges. I'd like to present some of our brain-gut work here briefly. And what we've shown in prior work is that TBI induces gut dysfunction broadly at the level of the colon, that may be malabsorption or dysmotility, but it also causes increases in permeability and downregulation of claudins and an increase in permeability within this system. What we also don't know very well is what's the effect of a secondary colonic inflammation or secondary challenge at the level of the gut that may actually impair the brain. And in the context of that, we know that secondary colitis can induce mucosal and inflammation. And the goal of Marie Hanscom's project was to determine what's the impact of delayed colonic inflammation on neurological outcomes. To do this, Marie took injured animals, and then at 28 days at this chronic time point, provided a chemical colitis model called DSS to induce this secondary colonic inflammation. Then she allowed the colon to recover and followed long-term neurobehavioral assessments. And what was most striking in these results, as shown here, at this 28-day time point when there is this secondary inflammation, you can see that there's a marked change in this motor performance. So those sham injured animals, which have a craniotomy alone, all of a sudden start to have motor function impairments when that secondary colonic inflammation occurs. Also, the recovery of the TBI animals seems to be altered such that they don't recover at the same rate. And Marie went on to do some, to evaluate the autonomic response. In particular, she used electrocardiography to look at the heart rate variability and the low frequency indicator of sympathetic tone. And what you'll notice here is in all those animals that receive DSS treatment, in the sham or the injured animals, there's an increase in this LF frequency, which indicates a sympathetic response, and this is associated with pro-inflammation. And when she evaluated inflammation broadly, she could see in the mesenteric lymph nodes and more broadly in other regions that there was an increase in these regions and an increase in systemic inflammation. To go back to the brain, what's the effect of this increased systemic inflammation? Well, Marie, at the level of the hippocampus, evaluated the inflammatory profile of these cells using these nanostring technology. And what she noticed is those animals, the sham animals that received DSS, had increased markers of neuroinflammation, disease association, and lipid metabolism, all these negative aspects that are associated with neurodegeneration. And when she quantified neurons in the injured hippocampus, you could see that there's a marked increase in this neuronal loss within this DSS group from the sham injured animals, and an exacerbation of the TBI effect with this treatment. So what does this show? It shows that prior to colonic inflammation, there's the normal traumatic brain injury response associated with motor and cognitive dysfunction and neurodegeneration. But if you have this secondary inflammation that's associated with systemic inflammation, under the conditions of sham and DS of TBI, you have this exacerbation of motor and cognitive function that's associated with microglial associated neurodegeneration. The mechanisms are many and varied, and we're starting to explore these, but may include secondary effects of the microbiome, immune cell infiltration, perhaps evolving from Th17 cells, trafficking to the brain to cause brain dysfunction. And these are ongoing studies that are of interest to many. So briefly, and to summarize, what we've shown is that chronic neuroinflammation can drive neurodegeneration and neurological decline, but it's not simply set to the brain-mediated effects. There are also extra brain effects in the periphery, and studies in the lung and gut suggest that these are indeed predominant. So I'm going to finish up there and thank the sponsors for this research, and also very much Marie Hanscom, Rebecca Henry, and Rodney Witzel, who have really been pioneering this work in my research lab at University of Maryland and more recently in Dublin. Thank you.
Great. Thanks very much, David. Beautiful talk, as ever. Time for a couple of questions. There was a question from Mario Forzioni. What's the relationship between blood-brain barrier damage and microglial activation or damage? So where does the blood-brain barrier fit into the story?
Yeah, well, in the context of these more severe injury models, there certainly is a damage, acute phase damage of the blood-brain barrier. In the context of chronic brain injury, it's less well studied, although neuropathological assessments suggest that there is chronic phase leakiness of the barrier, which then may allow these peripheral immune cells to continue to traffic to the brain. Chemokine signaling may still be in play. We believe, with regard to those secondary colonic inflammation, that the secondary challenge, that blood-brain barrier may be a critical driver of this secondary effects, because what you'll notice is the animals that had the sham surgery or the TBI actually had the vulnerability to that systemic inflammation, but the animals that did not receive any kind of brain surgery were resistant. So we are wondering, is there a blood-brain barrier component to this? And that's certainly something that Marie Hanscom is pursuing in future studies. So blood-brain barrier, key part of the story.
Fantastic. And just maybe a very brief answer to this, because we're kind of out of time, but from Barry C. Mungle, to what extent does the impact of TBI on motor/sleep function, rather than or in addition to the direct effects of TBI, influence microglia? So you know, these kind of more non-specific, I suppose, effects, you know, how would you control for that in the studies that you've shown?
Yeah, well, people have shown that experimentally that the increase in ICP is associated with changes in meningeal lymphatics and glymphatic responses, which then are shown to drive these secondary microglial responses. So how you control for these, you know, I think we have to be careful about how we perform these preclinical studies. It's important to note that all these studies were done under the correct light cycles, so that we were able to maintain all these aspects. But it is possible to do experimentally, but a very important caveat. I agree.
Fantastic. So there are lots of other questions actually. Perhaps you can put some of them in the chat, but we'll have to move on, I think. So thanks again, David.